The overall goal of the following experiment is to test the short and long-term memory of a learned positive association between food and the chemoattractant butanone using C. elegans as a model. This is achieved by first starving synchronized well-fed young adult worms for 1 hour. Next, the worms are trained in the presence of food and butanone by masked conditioning for short-term associative memory and by spaced conditioning for long-term associative memory. After conditioning, chemotaxis towards butanone is measured and compared to naive chemotaxis to study memory retention. Results are obtained that show the duration of short and long-term associative memory of the learned food-butanone association based on changes in the learning index.
This method can help answer key questions in the cognitive aging and disease fields, such as what genetic pathways are required for neuronal function in normal, aging, and neurodegenerative disease states, and how learning and memory can be improved under conditions of cognitive impairment.
Worms are prepared using standard methods on 100 mm high-growth media plates seeded with 1 ml of OP50 E. coli. Use young adults at a density of around 5,000 worms or less per plate. Collect the worms by gently pouring M9 buffer onto the plate and swirling the worms free. Try to minimize agitation, which may dislodge unwanted bacteria. Next, tilt the plate and pull up the buffer worm mixture from the corner using a P1000 pipetteman. Transfer the worms to a 15 ml conical tube, taking care not to pipette the worms against the side. If worms remain on the plate, repeat this step.